So today I will talk about causal representation learning of genetic perturbations, especially with emphasis on the perspective of identifiability and combinatorial extrapolation. I'll explain those two terms during my talk. So here is an outline of my talk today. So it will be split into half. For the first half, I'm going to give a short primer on causal inference. This will mainly cover some mathematical concepts that are relevant to understand the talk today. So it will not be a comprehensive primer on all of causal inference. So mainly I will talk about the definition of structural causal models, interventions in terms of structural causal models, and then I'll briefly try to summarize a few relevant tasks in genomics that we've been considering. And that's the first half of my talk. For the second half, it's going to be a research talk on my a recent paper in Europe. This is a joint work with people at IBM. For this part, I will first introduce the causal representation learning setting, and then I'll give a contrast to the traditional causal inference setting, and you will see why it makes sense and why there are difficulties to study them. And then I will cover the identifiability part of this talk, and then I will talk about algorithms, and then I'll show some results on perturb-seq data, and I'll draw conclusions. So I'll get started with the primer on causal inference. I'm going to give the mathematical definition of structural causal model. But before doing that, I do want to point out here that you can see on the right of the slides, here's a picture of theorized path out diagram path analysis. I want to point out that when I was talking about foundations of our work, trying to build something that understands genomics, and in fact, the foundation of this foundation, which is the foundation of structural causal model, actually is laid like 100 years ago by folks who are actually working in genomics. So for me, it's always interesting to see like how these two fields kind of grow together and working in this interdisciplinary area. I would usually try to draw inspirations from real applications in genomics and then try to summarize mathematical model out of it and see if we can actually develop new insights from causal inference perspective to understand genomics better. So with that, I'm going to give the definition for this. I'll be using this 4-node diagram to illustrate my example. So when you see structural causal model, it's usually always associated with a directed network that looks like this. In this directed networks, the causal relations are associated or given by the direction of this network. So what I mean by this is that you will know that each node of this directed network will be associated to a random variable. For example, there are 4 nodes here, so there are 4 random variables in this structural causal model. And then the edges actually entail the causal relationship between these random variables, in the sense that each random variable will depend on its parents in the directed network, and also some exogenous noise that's specific to this endogenous, sometimes called endogenous variable. And usually the exogenous noise variable are assumed to be independent. So concretely, you will sometimes see equations that summarize the structural causal model in the form, for example, for this 4-node graph like this, where you have x1, which has no parents in the directed networks. So it will only depend on the exogenous noise variable to itself. And then it will be some G1 is its mechanism of generating x1. X2 as well, it doesn't have any parents. So it will just be a function of its own exogenous noise variable, epsilon 2. And then x3 will be, since it has 2 parents in the graph, it will be a function of x1, x3, x2, and also its own exogenous noise variable epsilon 3. And you have the same thing for x4. I would also like to point out that this mechanism or structural causal model are sometimes implicitly defined in the Markov factorization of their joint distribution. So sometimes people won't exactly put out these equations, but they would write out the joint distribution between all the system variable x1 to x4. And then you will see a Markov factorization, which basically means you would first have the distribution of x1 and x2, and then you will have the distribution of node x3 depending on its parents, and also the distribution x4 depending on its parents. So if you contrast this factorization versus this system of equations, then this G1 epsilon is basically summarized in Px1. And then this x3 equals to G3, x1, x2, epsilon 3 is summarized in P x3 given x1 and x2. So I also want to point out that this is defining the observational distribution, or sometimes we call it control, which means basically the system of variables that hasn't been perturbed or intervened. So any questions regarding the definition of structural causal models? Very cool.
Question. So in the context of cells, these would be genes you would use as genes.
So in the context of cells, in the fully observed setting, x1, x2, x3, they could be genes. And since they're random variable, they could define some measurement of the genes. But then in the CRL setting, you will see a different thing, which is they can be programs that are latent. So with that definition, I want to try to define interventions, which sometimes can be used to summarize genetic perturbations. So SEM, defined on the previous slide, gives us language to describe interventions and how the system changed under interventions. So what do I mean by that? I can give an example where, for example, I'm intervening on X3. So what this means is that it will change the mechanism that generates X3. So if you relate it to the equations on the last slide, it basically means I'm going to go into my SEMs and change the mechanism that generate my X3. And then this will have a downstream effect on node X4 because the distribution of X3 is changed. So you also see like this can be summarized in the Markov factorization as well by just changing the mechanism of that generates X3. Since I won't be talking too much about counterfactuals, but I will just give a definition of counterfactual here to see how it contrasts with interventions. So usually when people talk about counterfactuals, they think about questions such as what would have happened to the system variable if I had done some intervention I, whereas intervention is what will happen to X if I do I. So the difference in mathematical forms of counterfactual versus intervention is that after you do intervention, you can just look at the interventional distribution as itself. But then when you talk about counterfactual, you're actually thinking retrospectively in the sense that you will have already some observation of X. This observation of X will come from a set of exogenous noise variable, which will dictate all the randomness in your X. And then when you talk about counterfactual, what we mean is that we will fix the exogenous noise variable, and then we change the mechanism so that the counterfactual sample and the factual sample, they will share the same exogenous noise, but they are actually generated from different mechanisms. So that's a way to think about counterfactual in this SEM perspective. So I would like to point out that another way of thinking about the observational distribution defined on the first slide, the intervention and counterfactual defined on the second slide, inside Pearl's letter of causality, where you see that he has 3 layers. The first layer is association, which means that you are only doing something by seeing or observing the actual world. And the second layer where you give interventions is the case where you're actually actively performing something in the real world. And then the counterfactual would be imagining or retrospective things. So in some sense, we'll never be able to get counterfactual samples in this one universe. So Pearl defined this ladder of causality in the perspective of artificial intelligence because he's actually an AI researcher by born. So for us, I think we can also think about how single cell data for genomics plays a role, how to interpret that in this ladder of causality. So what I mean by that is that for the association level, you can think of it as measurements of cells that are in the control population. So for example, this can be just single cell RNA-seq without any perturbations, or it can just be some samples drawn from some patient's tumor samples without actually performing any treatments. Whereas on the second level, where you have interventions, for example, you can think about it as single cell perturb-seq, where you perform some perturbation onto the single cells, and then you get the measurement of those. And then for the counterfactual, what you actually want is that you want the control cells, but not in themselves, but like imagining what would happen to them if you actually perform some intervention on them. So in some sense, we're never getting access to such data because one is that we're never going to see the same sample at different state at the same time in both form. Another set is that if we do single cell RNA-seq is actually destructive. So you will never be able to observe perturbed cell before the intervention. So some other types of measurement, you might get some proxy of the counterfactual samples, but you have to assume that the time is static in order to assume that's the counterfactual sample. So that's basically a definition of interventions and structural causal model. And then on this slide, I'll just briefly summarize 3 relevant causality tasks in genomics. This is not exhaustive. There are a bunch more out there that are very important and interesting to study. But I will just summarize a few to give us a sense. So the first one is what we know as causal structure learning. So how that plays out in genomics, you can think of it as learning gene regulatory networks. But in the sense that when people usually talk about causal structure learning, they talk about learning the structures between the variables. Whereas in gene regulatory networks, you might also be interested in learning the mechanisms themselves, whether it's a downregulation or an output regulation, things like that. So I won't talk too much about this, but I do want to point out that a lot of the current algorithms in causal structure learning, they are very limited by the scale of data they can handle. So when you think about the case in single cell data, you actually have maybe more than 20,000 genes. If you want to learn a large network of that, it's really difficult. So this part I won't talk about too much, but you will hear from Romain's talk in the second half about a very nice approach towards this. This is the first task. And then secondly, which is going to be the task that I will cover, is what I would frame it as extrapolation to new interventions. So what I mean by that is that, for example, you have a measurement of cell by gene matrix here. They can be in the control population, or they can be under certain perturbations. And what you want to do is predict what happens to the cells, a cell by gene matrix under an alternative novel perturbation. It can be a perturbation combining the perturbation existing, or it can be a novel single gene perturbation. So this is predicting the effects of genetic perturbations on the cells. And then I won't mention it today, but there are recently there's also interest to do experimental design in the sense that you want to find optimal perturbations. So for example, you would have a source cell state, for example, fibroblast, and then you have a target cell state, for example, iPSC. And then you want to find like the set of genetic interventions such that it will move your cell from the source cell state to the target cell state. So for that fibroblast and iPSC, you can have Yamanaka factors. But then for other pairs of cell state, this is less well understood. So if you can come up with experimental design to test combinations of perturbations in rounds, then probably you will have a better chance of finding something that gets you closer to the target. So this is all of the tasks I want to summarize here. And now I will just go to...
Jiaqi, before you move on, I have a quick question about the previous slide. About predicting the effects of genetic perturbations. But like, how do you think of that in relation as a causal task in relation to like it's a prediction task, like a classification task or a regression task? Because I mean, here you're saying that you somehow by incorporating causal ideas can do better at prediction.
Yeah.
Is that what you...
Yeah. So I want to give a high level way of thinking about it without getting into the networks and all those assumptions about the causal graph. So I think if you think about it as purely prediction, you will actually treat the control population and perturb population separately. And what you want to do is just try to predict what happens in a new environment. And then you use some feature of the perturbations, for example. But if you think about it causally, the perturbed distribution is the control distribution under the perturbation. So you kind of have this link that the perturbation is changing the control distribution. So if you do this, you can, for example, you will see later on my approach is taking the control cells and then try to predict what happens to them after the interventions. Whereas the pure prediction, you can just directly go to the perturbed distribution.
Key thing is modeling the relationships between distributions.
Yes.
That's what makes it
yes.
causal.
That's the key thing without getting into the network structure assumptions. With the network structure assumptions, there's more things to care for. Okay. Yeah.
Thank you. Very general question. So how do you construct the causal graph?
How do I contrast the causal graph?
The structure of the causal graph.
Yes. I will give some intuition on the next few slides. I'll give some intuitions. Yeah. So now I'll just move on to the talk about this causal representation learning paper that we have recently. So I will first start by describing the CRL setting. CRL is just a short abbreviation for causal representation learning. So before talking about that setting, I do want to give an intuition of this, of the like the traditional setting where we fully observe the causal variables. And I'll give some intuition why the structure, how can the structure be learned. So this is not the CRL setting, but this is the setting preceding CRL, which motivates the CRL setting. And then you will see the contrast later on in the next slide. So in this fully observed setting, what I mean is that I have all my causal variable in this causal diagram observed. What I mean by that is that I can actually have measurement of each random variable here. So you can think of a very tabular-like data set. So the intuition of why the causal diagram is learnable in this setting is that the structural causal model actually entails a set of conditional independence relationships. So if you look up in literature, this is sometimes described by D-separation arguments. So it's a set of arguments that tells you if you have data following this SEM, what are the potential causal, conditional independence relationship in the samples that you observe would be. So for example, in this diagram, this grouping of X1, X2, they are independent of X4 given by X3 because X3 blocks all the paths from X1, X2 to X4. And then you also notice from this diagram that this separation will tell you x1 and x4, they're not conditionally independent. So thinking about this, what this actually means is that if you have the SEM in a black box, you don't know what it is, and then you have samples drawn from the SEM. What this will tell you is that in the samples that you can observe, there are some conditional independence relationships that are indicated by the black box causal graph. So this is what I mean by graph actually point to some conditional independence in the data. But then when you want to construct a causal diagram, we want to know what the black box causal diagram is from just the data. You need the reverse direction, which is data to graph. And this is given by the faithfulness assumption, which basically means there's no additional conditional independence in the data, aside from the ones that are entailed by the graph. So what this means is that all the conditional independence relationship you see in the data would be some form of graphical relations in the black box model. So given this assumption, you will have the reverse direction of data to graph. So I want to point out that this assumption, this is like debatable whether it's strong or not, but you can probably show in some settings, under some assumptions, this assumption will have measure, like violation of this assumption will have measure 0. So if you're given this assumption, then what you can do is from the data itself, you can infer something about the graph. And that's how you would usually construct the causal graph. Won't talk too details about how that's being done, but then this is the intuition why it can be done. I also want to point out that what you can do with interventions is that, for example, if you intervene on X3, you will know that by the definition of interventions, if you think about the equations that I put out in the first 2 slides, you will notice that an intervention, it can only change the marginal distributions of its downstream variables. And in this example, for example, is that if you intervene on X3, it will not change the distribution of X1 and X2, but it will change the distribution of X4. So this is also something that the graph can tell you about the data. So if you compare the interventional distribution to the observational distribution, you will only be able to see the changes in the downstream variables. And then what you want to do is also like data to graph, this reverse direction. So this is also another faithfulness assumption, oftentimes called the interventional faithfulness assumption, which basically means I assume that all my downstream variables, they will have their distribution change if I perturb something upstream. So with this assumption, the data to graph direction can potentially be more refined compared to the observational case. And this is why with interventions, you can learn more about the causal graph. I hope this makes sense. So I'll pause here a little bit to see if there are questions. Okay, no questions.
I have a question about how did the arrows in the graph relate to like time? Is it, you know, when you think about like stuff happening in a cell, you typically think, or you can think about it in terms of time. Yeah. So do you think of these relationships in the graph as having some time and yeah?
I'm usually thinking about this at equilibrium. So like time series causal inferences, I guess less well understood. And then if you have that, I think what happens is you can have a set of distribution for time t minus 1, and then you can have the same set of variables at time t. There are some links between the variable at this point and this point. And then you can somehow draw a causal graph at the equilibrium state, but it doesn't have to be an acyclic graph.
Framework be extended to weighted networks, e.g. the two different distributions may differ only if the level of the perturbation is above a certain level.
Yeah, yes, you can define that just by defining the perturbation only changes the mechanism if the level of the strains is higher than something.
Thanks. I just wanted to follow up on Salil's question about time. So if something is causing something, I guess something happens first and then something happens after the first thing. So should I view the tree as having time? Right? Is there a sequence? Like what should I picture time in that sense?
I think that's one way, but when I was answering to Salil's question, I was pointing at another way, which is you observe the same set of system variable across time, and then so imagine PDE, you have x at time t minus delta t, and then you have x at t. And then if you do the derivative of your x versus the previous x, then you will see that some entries of your current x at time t will depend on some x of the entry of t minus delta t. And then if you want to summarize all of this with just one causal graph with the same number of nodes as your x, then you can try to summarize this process in one causal graph. I think what you're talking about is I create a separate set of random variable for each time point.
So are you saying that I should view this graph as representing the derivative of these things because it's all at one instant, but it tells you what the flow is going to be?
Yeah, I think so. But this is, if you view it in a time series data, this can be a little bit restrictive in the sense that if you summarize all of them, it doesn't have to be an acyclic graph. It's directed, but it doesn't have to be acyclic. You can have self loops. For example, for x1 is actually is realization at time point t depends on its realization at time t minus delta t. So then you will have a self loop here, which I'm not covering over here. At equivalent, yeah. That's a good question.
Is the model that you do an intervention, let the thing get to equilibrium?
Yes.
Then you have this as the acyclic approximation to whatever happens in that system, even though you'd expect there to be some like.
Yes. So what happens is that all of this is summarizing the equilibrium state. And then all those joint distribution is also summarizing some equilibrium distribution. Yeah, I think that's the case.
So as a simple example, if you set something to 0, then at equilibrium, it's going to be 0 as well. So if you set like x1 to 0, then let the system go to equilibrium, then it's going to be still be 0.
But sometimes you have feedback loops, so you would have cycles here. But I really don't want to get into that.
Right. Okay. Yeah.
The cyclic. I want to point out another thing about the acyclic assumption is that like people usually put acyclic conditions on or assumptions on this because under this assumptions, this system variable is can be uniquely solved. So that's like one other reason why people usually put acyclic assumptions. I guess that's like a less well-defined, and I'm too sleepy to try to think about how to define it now, but we can talk offline. So I will just move on to the CRL setting then. So in the causal representation and learning setting, you will have a stark contrast between the fully observed setting in the sense that everything about the SEM is actually hidden in the latent space. So what I mean by that is that the causal variables themselves and also the structure between them, they are latent, and we only observe them through an unknown mixing function. So you can think of it as I have some hidden regulatory elements and network here. And then I have some pictures of the cells that are somehow, I would imagine as some manifestation of the latent regulatory relationships, or you can have a very high dimensional single cell data set cell by gene matrix, where it's actually generated from just a few gene programs. So I do want to point out, like, aside from a philosophical perspective, such as which one of this model actually describe the ground truth, there are some pros and cons of the causal representation learning framework compared to the traditional fully observed setting. So apparently, in this setting, you can have a much lower dimensional causal model in the latent space. This, in some sense, might be easier to learn, in the sense that you don't need to learn a network between all of the observed random variable, which can be 20,000 dimension in the single cell data, for example. And then another benefit of this setting is that it can incorporate any flexible mixing. So this would allow you to be able to handle beyond tabular data. For example, looking at images. Looking at images, you cannot do causal structure learning on top of the cell because it doesn't really make sense to treat each pixel as a causal variable, because if you compare the images across the data set, those entries of the image, they don't actually correspond to each other. They don't have any relationship. So there's also a few downside of this framework, in the sense that since everything now is hidden, it will be harder to establish any theoretical guarantees of identifiability. In the previous setting where you have the causal variables themselves, the identifiability question is how do you uniquely learn the graph? But in this setting, you will need to learn the causal variables themselves as well. And also, the interpretation of the learned variable and structure in the latent space will have a harder, you will have a harder time to interpret those because these are actually latent variables that you learn instead of just the observed variables, where you can easily interpret what the causal graph means. So with that, I'm going straight into the setup with mathematical definitions. So here's the setup that we are considering in CRL. So this is not the unified setup. This is just a setup that we start with to establish this work. So in this case, I'm just grabbing a random cell by gene matrix here and do some illustration to illustrate the setup. In here, I'm given unpaired data sets from observational distribution and interventional distribution. If you think back on the first few slides where I defined this observational distribution, it can just be cells from the control population. For example, targeting, non-targeting inside my Pritchard data. And then the interventional distribution, they can be multiple, where each one of them would be targeting, for example, some target gene GI. And this can be, for example, I'm targeting ATS1. I have observations of X space, which is put here as a diagram here. And then my assumption is that this cell by gene matrix is actually generated from a latent causal model between variables where I denote as U. And the latent causal graph is denoted as G. I'm assuming that for both the observational distribution and the interventional distribution, they share the same mixing function F. For those samples in the perturbed distribution, they can be generated via another distribution of the latent variables, but they have to share the same mixing versus the control population. And then you will notice that I denote also the perturbation or the intervention in the latent space as I. This will also be something that I need to learn, in the sense that I'm giving you only a few data sets as this G1 up to GK. And now what you want to do is you want to learn what those distributions correspond to, in the latent space. So somehow, aside from the GI here, you also need to learn what are the targets and what are the values this perturbation sets the latent variables to. And that's why I have an I here. And then the goal here would be to identify everything in the latent space, including the causal variable themselves, the graph between them, and also the intervention targets. So the question I have here is, how do you actually interpret this latent things? So for example, you can try to interpret the learned eyes as groupings of target genes. So there are actually groupings of target genes. You can try to interpret them as, for example, programs of targeted genes. So this will be just a set of target genes that correspond to the same program because they act on the same latent variable. And then you can try to interpret, for example, the causal graph between the intervention targets here as the regulatory relationship between programs. And then for the corresponding entry U in this latent case, this will be just a scalar value. So you can, for example, because they correspond to the eyes here, so you can interpret them as, for example, some program score. And this is like one interpretation you can do in terms of the latent graph, latent causal model that you identify. And then aside from identifying these variables, you also maybe want to learn a generative model that allows you to produce cell by gene matrix for a new intervention G, given the control population. This will then allow you to do, I think, statistical perturbations at the single cell level. So this is very fine-grained or mathematical definition of the setup I'm considering. I'll pause here a little bit to see if there are any questions. Yeah, can use my yes.
What is curly G?
In the perturbative example, it can just be the target genes.
It seems like it's a distribution, right?
Yes. So G is a distribution, but DG is a distribution. It's a bunch of samples from the distribution, but G is a superscript indicating which distribution this is.
But so you have U coming from G.
Oh, this G here.
Yeah, curly G. I call it curly G.
Yeah,
sorry,
no
big
worries.
G.
Curly G is the latent causal graph. So I didn't really put out the distribution PG here just to say for simplicity. So G is the graph, the structure here, and then you will have a distribution that factorized according to G.
We have a question in the chat which says from Fred, I guess expanding on how you interpret the latent features. How do you conclude that a latent feature is a pathway or something biologically important like that?
Yeah, I don't think I can conclude that, but this is just one way to interpret it. And then you can possibly do some downstream analysis to further verify whether it's actually a pathway or a novel program like that. I'll then talk about identifiability and algorithms in this setting. But then, like before talking about that, I want to just like high level, what should we think about? What are identifiability or algorithms? How should we think about this in like the general sense? So when I was talking about identifiability, usually in CRL setting, there's some latent causal model here, which you don't get to observe is in a black box. And then what you observe is some realization of coming out or mixing of the latent causal model. That's your observed data. So what identifiability is a question of going from observed data to latent causal model. And the question usually being asked is that, is the latent model that explains the data unique? So usually when you think about identifiability, it's trying to answer this question, or is it unique up to some trivial transformations?
Does this uniqueness refer to assuming that the data is generated by the model?
Yes. Or
uniqueness assuming that, just given some data?
The uniqueness is assuming that the observed data is generated from this class of latent model. And I'm trying to answer is from this class of observed latent model. Sorry, from this class of latent model is the observed samples uniquely coming from one of them. And then the algorithm perspective is how do we learn the latent model from the observed data? So this is generally how I like to think about this. So I'll just directly go into the identifiability result. I just put out some proof sketch in case anyone in the audience is interested. For the theorem itself, it basically says that if the interventional data from at least one intervention per latent node is available, and I have some faithfulness assumption, then I can identify uniquely the latent interventional targets and also the causal graph between the interventional targets up to trivial transformations like permutations. So we will notice that the latent variables U themselves, the scalars themselves, they're actually not identifiable in this setting if you are given a very general class of interventions. But then if you restrict the interventions to like due or perfect interventions, then your identifiability of U can also be guaranteed. So I put out a proof sketch here in case the audience are interested. I'll just quickly describe this. So in order to get the identifiability of the latent variables, in the first step, what you would do is you will use the data you observe to actually identify some linear transformation of the ground truth interventional data U. And this is given by a constrained set of mixing function and assumption on the non-collapse support, mainly from this Aguirre paper from 2023. So this first step will give you some U hat that you can identify, which will be a linear transformation of the ground truth latent variables. And then in the second step, what we would do is that you would actually refine the identifiability by identifying a topologically ordered representation gamma. So what I mean here is that in the second step, you will refine the linear identifiability here to the case where gamma is an upper triangular matrix. So this is given by enforcing that the intervention can only change its downstream variables, which I sort of try to give a bit of intuition in the fully observed case. So this is a little harder in the CRL setting, but that's basically how you get to the topological ordered representation gamma. That's the second step. And the third step we will do is that inside all this upper triangular gamma, you will try to enforce that the number of changes to your latent variables are minimum. Then you will get a very sparse gamma, which is diagonal. And after getting gamma to be diagonal, you basically observe the latent variable up to some affine transformation. And then it's almost equivalent to the fully observed setting where you can just treat your identified hat U as a causal variable and try to learn the structure between them. So this is a brief summary of the proof sketch. Question? Can you use the mic? Thanks.
Yeah, so how do you have to decide how many latent nodes you want?
Yes, you have to decide how many latent nodes that you want, but that's not an assumption. That's given by the first linear identifiability. So if you assume non-collapse support, then by observing the data, there's actually an inherent minimum dimension that you can describe this. And that dimension will give you the number of nodes in the latent space.
So practically, how do you find out the dimension?
So practically in the algorithm, we just set it to some value, and then we just, in order to learn a graph, we just reduce that value until the intervention targets, they don't collapse in the latent space.
Oh, and another question, how do you know that you have one intervention per latent node?
Yeah, that's an assumption that we cannot go beyond in the current development of CRL, but we're working on multi-node scenarios.
Just following up on Shini's question, can you give like biological intuition for what the latent nodes are? Is it like, are we supposed to think of them as like almost like the programs of the cells?
Yes.
Similar to like when you do other dimensionality reductions like PCA or those lines?
So I think mostly I'm thinking about the latent U, the latent causal variables as programs themselves, because I have like the interventions on this latent node, and the intervention target I learned is actually some groupings of a bunch of target genes. So I'm thinking about that as programs.
So like for your theorem, then it's not like you need 20,000 interventions, it's you need one intervention per program.
Yes, for all the relevant programs that you generate. That's the only thing you can learn. Yeah, as of now.
So biologically, it seems really hard to do a strong intervention on a like cell program, or gene program, I don't know what you were calling them. But so like, this identifiability result requires hard interventions, or can it have a soft intervention?
Soft.
Okay.
Any intervention. So
Okay.
that's what I was saying, that here you don't get identifiability of U themselves by assuming a general class of interventions. But if you actually only consider hard or perfect intervention, you have stronger identifiability results.
Okay. So the idea is you kind of are doing some sort of intervention. You don't, I mean, it's hard to think about like doing an intervention on a variable that you don't know exists.
Yeah.
But you're just assume that you're doing some intervention, and that hopefully is acting as an intervention on the latent space in some way.
Yeah.
Software.
So I would agree that the single node intervention in this scenario is a bit restrictive, but if you consider multi-node, then it can actually be anything. So you can have chemical perturbations, and it's acting on a lot of the latent programs or latent variables.
So this allows multi-node or no?
This doesn't allow
single
so
okay.
far. Yes.
Thank you.
I don't have a summary of the pre-existing work because of time restrictions, but I'm happy to talk about that offline. So yeah, basically multi-node is unsolved. Okay, cool. There's a question in the chat: how much data is required to train the model? Yeah, I will then describe. I'll give a little bit comment when I describe the algorithm. So I'll just quickly go through the algorithm. We call this discrepancy-based variational autoencoder. So what this algorithm does is that it takes in X, which is the control, which is, for example, a control sample, and it takes in a perturbation label, for example, what target gene you are perturbing. This is G. You put them both through two encoders that are separate, where from X, you learn the exogenous noise, Z, and then from G, after the encoder, you learn what is the intervention on the latent node is, that's I. You put both Z and I through a causal graph. This will give you the intervened version of Z, which actually essentially will be the causal variables themselves. And then you put through the decoder, this U, to generate your perturbed sample XG. So the causal graph is actually coming from this latent scenario where we are enforcing that an intervention, for example, if it's learned intervention I is acting on Z2, then it will only changes your intervene Z after this causal graph will just be changing these three entries of Z that are downstream of I. And then another point I want to point out of why we're calling it discrepancy based is because think about training it where you are feeding in a control sample and some perturbation. And this is like the perturbed version of your control sample that you generate. So if you want to like train this with real data, you can not actually compare that. I'm sorry about the illustrations. So sometimes it's supposed to be a cell in another orientation. But I don't know why it's text now. I tried to fix it last night, but it's coming back. So my point is, this perturbed sample, you cannot just compare it with a random sample from the perturbed distribution because a random sample from the whole perturbed distribution will not correspond to the exact control cell you put in the variational autoencoder framework. So this is not directly comparable. So what you want to do is that you generate a bunch of perturbed samples and you compare distributionally this fake counterfactual samples versus your perturbed data. And then that's where the discrepancy-based loss is coming in, is actually indicating that you're using distribution metric to train the whole thing. So I already talked about the loss function, which will, aside from the standard variational autoencoder loss, you will have this discrepancy loss between distributions, which we use maximum mean discrepancy, and then you have some sparsity on the latent graph that you learned. So the overall schematic would be that you input some control samples from the unperturbed version, you input some perturbation label, then you get some virtual counterfactual samples. And this is more finer grain of the verse or schematic. It's also in the paper, so you can look it up. And I'll just quickly go over the results on the Norman data set. which is a data set consisting, I believe, 105 single node intervention and 133 double node interventions. So to learn the programs, we just like reduce the dimension down until it doesn't collapse anymore. So it's roughly 7 programs that we identified. We didn't do much of more interpretation into the programs as of the stage of this paper now, but it will be interesting to do more finer grade analysis on the learned programs themselves. So do you want to point out that since it's a variational autoencoder, it does allow you to generate some data from the single gene perturbations. So these are some results on the left out samples, KLF1, C, PB, E, and you can see that the actual cells, which are in blue, they kind of overlap well with the orange generated cells. You can see it more clearly by contrasting the actual cell versus the generated cell. Okay, so these are UMAPs. So I think everybody's familiar with that. Sorry, I didn't describe it. You can see that they actually look very similar. So this is for single cell perturbations. So there's some overall statistics as well, the MMD loss and also the R square and RMC between the means. I do want to point out another perspective of this is that you can actually do extrapolation to combinations of single node perturbations in your data set. So what happens with this is that instead of putting through one perturbation label to the perturb encoder, you put multiple, and then you will get multiple latent I's. And then this will allow you to generate counterfactual samples if you perturb the combination of these genes. So then in the latent space, what happens is that you do some additive effects of all the perturbations that you input. This will give you a zero-shot way of generating multi-gene perturbations. So for example, I have some visualization here. I'm never given the data any combinatorial perturbation samples. And somehow the model is able to learn some additive effects. For example, for ETS2 plus CPB1, CPB is quite overlapping here, and for this one as well. But then for certain combinations, you will see that there's a huge discrepancy. And this is largely due to because their effects are actually not additive in the latent space. And then here are more examples where you can see, for example, this one aligns well, and this one doesn't really. So I have all of them on Google Drive, I believe. There's the overall statistics. Somehow you can prove also theoretically that if you actually identify the latent graph, then that procedure I was telling you to generate multi-node intervention will give you exact distribution in theory of the combinatorial intervention. So I think I'll quickly wrap up now. Sorry for taking too long. So what I talked today is a bit of a primer to describe the relevant mathematical concepts. And then mainly after that, I talked about how CRL setting is different from the fully observed causal inference setting, in the sense that it can handle perceptual highly dimensional data, but then it will be harder to establish identifiability. And in some sense, this also calls for new algorithms and interpretations to learn the latent graph or latent causal model. And then the second thing I talk about is provable identifiability guarantees under single node interventions. So you can think of extending this to multi-node intervention, which will allow you to handle more general scenarios. And then the third thing I talked about is the identifiability guarantees for that combinatorial extrapolation, very briefly at the end. And then also how we learn it using discrepancy-based EAE. So again, here's the link to the paper, and we also have our code base available online if you want to try it out on your own perturb-seq data. I think, yeah, that's all my presentations.
Today, I would like to talk about scaling up causal discovery approaches. And this is work I did at the very beginning of my postdoc. Before getting in further depth, I would like to give some kind of introduction and introduce myself, I guess, as a scientist. So biology to me is a study of living organisms. And we're usually interested in doing this at several scale, like a population or an entire organism, tissue and going down to the basic unit of life, that's the cell. In order to study this, researchers started making assays, experiments, and technologies to either corroborate or refute biological models. As this assay became high throughput, we started relying on statistical machine learning to analyze experimental data. And somehow these days we're envisioning that machine learning can also help in as designing future experiments, experimental design. So this is kind of like the basic premises of biology that I'm really excited to try to take a part of as a scientist. During my PhD work, I've been very interested in building scalable and statistically motivated algorithms for analysis of databases of transcriptomics data, single-cell RNA-seq and multimodal and spatial transcriptomics. Then I was also very interested in developing machine learning methodology, something that you can loosely call ML for science. So I worked on disentanglement, on Bayesian decision-making, and applications to molecular biology in general. I spent quite some time working with other people on designing and maintaining an open source package for making Bayesian models for multiomics data. So some of you may know SCVI tools. Something I did also as part of my PhD was actually to work with technology companies in the context of marketing and thinking about some of those offline counterfactual inference algorithms for recommender system. And something that left me on my hunger at the end of my PhD was, oh, that would be really great if what would be some kind of equivalent to some of these techniques in biology? What do they mean and what can we do with those? And that's what made me want to really excited about doing a postdoc. And so some work direction throughout my postdoc, a lot of my postdoc work is basically centered around perturbations. One first family of work is a building mechanistic model of gene regulation. That's what I'm going to talk about today. That's why it's in blue. And I'm going to talk about scalable inference methods from genetic screening data. I've also been very excited about, in general, learning what I call desirable representations. So that's a very loose word for and saying that we want to learn representation that respect the semantic of the perturbations. And usually that goes through either some thinking around disentanglement or causality, and sometimes those two things are actually pretty related. But that's not what I will talk about today. And generally, I've been really excited in this postdoc by being both at Genentech and Stanford to be able to collaborate with technology enthusiasts, but also with immunology specialists, still in the field of perturbation biology, but I've been very excited about high throughput chemical screening platforms, optical push-screen technology, immune cell activation in general. I've worked on T cells and active learning or experimental design. So if these are topics I would be really happy to talk about with the people I meet later today or in general. And so for the purpose of the talk today, I'm going to first introduce what I mean by gene regulation, we've already covered some of these topics, but I'll still go through them quickly, talking about perturb-seq, about causal discovery learning, causal structure learning, and then I'll present the paper that is a bulk of the talk, differential causal discovery of factor graphs, and finally I'll give some closing remarks. So let's get started talking about gene regulation. Gene regulation is difficult because it involves intermediate proteins that are binding and that are usually not observed in those genomic data sets. So here is what I mean by gene regulation. An example with growth factors. So we have those molecule called growth factors that are binding to the receptor of a cell, and upon this binding, the receptor can phosphorylate or do some other chemical alteration of something called the transcription factor, that green molecule that's going to then move back to the DNA, bind on the DNA, and activate transcription of different genes. So here, if it's a growth factor, usually we want it to activate division promoting genes as well as growth promoting genes. Those RNA can be translated and we obtain proteins, and some of those proteins can actually be transcription factors and go back to the DNA. And that's usually what people mean by signal transduction or gene regulation. So and that's what I will have in mind as a mental model for today. So this is a fascinating field and a very difficult one. People usually think about as kind of questions, what genes regulate a certain cellular phenotype? And that's what those screens are meant to be, to find those regulators. Once you find regulators, you can start asking about mechanisms. Why is it that something regulates something else? And if we can understand all of those questions at the biochemical level, then we can start finding good targets, and some of them might be druggable, and we can develop drugs. And that's why people are excited in general. And those are all very hard questions, and the answers will not be on this talk, on this slide. So, but some technologies that got invented for the purpose of not solving, but answering some of those questions at least, is the pooled genetic screenings experiment. So in those experiments, we're able to, in a bunch of cells, we take a bag of cells, and in all of those cells, we can have a different guide RNA. Those cells have been meant to express Cas enzyme, so they can act on the DNA. And with a different guide, you can act at different places in different cells. That's why they have different colors on the left. And then there is some kind of molecular biology engineering to be able to understand after sequencing which perturbation or which guide was in which cell. And we can also run single-cell RNA sequencing. This is all done at the same time. That's called perturb-seq, and we obtain also the gene expression level. This is the data we've been talking about also in the primer. So in this context, for every cell, we measure two things. First, what is the gene expression level? And second, which perturbation was in that cell? Is that clear? Yes. So what we hope to learn from those experiments is first, we can read this directly. What is the response in gene expression to a genetic perturbation? When I say directly, it's after cleaning the data, and that takes some time, but that's not, I'm not gonna give a lot of comments on this part. For example, I knock out a certain gene and I see all of those genes going up or down, up-regulated, down-regulated. So we also get the direction, right? And somehow what we hope to do is figuring out what's more direct and what's indirect, what sits in the pathway, in the way of regulating other things. And that can be a bit tricky because, as I've shown before, gene regulation goes through all of those proteins, and those proteins can be dimerized, can be phosphorylated. So many things can happen to them, and we don't measure that. So somehow even this notion of direct and indirect can be quite fuzzy based on the variable that we're actually observing. This is kind of the best I can say as an introduction so far. So what have people been doing as a vanilla way of analyzing those data sets? Usually people run flavors of differential expression or regression. So here is a regression. We categorize the perturbations. So after we process basically the matrix I've shown you, you can say in that cell we have this perturbation and here is a gene expression. We can fit that into a regression model based on looking at the effect size of changes between the perturbed cell and the control cell. We obtain those beta, those people call that regulatory matrices sometimes, and we can filter it, do a heat map, and do some clustering, and we can look at all of these perturbations are grouped into gene modules, and they affect downstream genes are called grouped into gene programs. So we have that's the semantic that was used in this first perturb-seq paper. And we can look at these groupings, and we can say, those modules are affecting those programs. Something that's kind of interesting to note for me was that in this first data set, people perturb transcription factors. And when you look at, so those modules will mainly be TFs. But when you look downstream at the programs, you also see sometimes the same transcription factor. For example, IRF4, I think, appears in M4 and program 2. Which is somehow expected, but is also some kind of thing we would like to, that shows the limitation of this approach. So it's a nice way of reading the data. It's kind of the output of the experiment. But maybe there is a better way of interpreting this data by thinking about gene-to-gene the graphs, right? So we would like to know that gene influences that other genes. And for today, I'm going to focus on acyclic graphs. So we know it's not true because you can perturb a gene and you see downstream responses. And some of those genes, when you perturb them, will also have an effect on what we thought was downstream, but it's actually upstream. So it's into their cycle in these data sets. and in general in those screens. But there are different ways to think about it. First, there is the mathematics that's much more complicated to figure out. Second, it's just a different kind of models. And actually, we have extended some of this work for the context of cycles, but I will not talk about today. And I will just say that somehow, if you do your experiment in some kind of short time scale, you have less of those effects. That's maybe one way to think about it, that the acyclicity is valid in some time scale in some systems. But that's kind of the best I can say. And I will take questions offline regarding this topic. So what do we get from this directed regulatory model? First, we can call that a mechanistic model because we get which gene influences which one. And ideally, that's already interpretable as such. Second, we get a set of hypotheses that can be tested in follow-up experimental work. Ideally, we can show this to an immunology expert or a biologist, and the person can say, oh, that edge is actually something that we didn't know before. The story is a little bit complicated. I'll show you real data, but ideally, that's what we would like. And in principle, once we have those densities, so we've seen in the talk before some examples of manipulated densities or interventional densities, where we could be able to predict the effect of interventions in any genes in the network. And this is, again, I write in principle because with the current method, we're not actually able to do that, for example. But yeah, that's why we would like somehow this kind of directed regulatory model. So, and why is it difficult? So first, the data is large scale and high dimensional. So we can have up to millions of samples in the recent Perturb-seq data sets. We want to consider at least thousands or tens of thousands of features, just considering genes. And we have many interventional regimes, meaning that we have many perturbations. Other complications are that we have measurement noise and confounding factors. Measurement noise, you can think about it as a stochasticity of gene expression and all of those things that you would like to consider. And for the purpose of the talk today, I've worked on this a lot in my PhD. And so thinking about all of those conditional distributions for generative models was very important for fitting the data well. And there are some efforts to be made in that end to make it work in this context. But this is not what I've worked on for the context of these slides and this presentation. But one thing that I've really tried to put some thought on is the fact that there is specific structure to be taken into account. The fact that many gene perturbations can have a similar effect on downstream genes. And so somehow there is some redundancy on how perturbation can affect things downstream. And we would like to come up with a model that can somehow fit a causal graph in that regime. Any questions so far, that introduction? So now I'm going to talk about causal discovery learning. So we've talked about those a bit. So I'm just going to say here, to make it clear, I'm not working on latent variables. I'm working on observed variables. So here, when I say A, B, and C, those are 3 genes. This is a graphical model. So there is not yet here any interventions. But basically, my graphical model is encoding a joint distribution. And we've seen some of those factorization. Here, I'm putting the structural equation model. And when you add causality, it becomes a structural causal model. So here it's kind of a linear Gaussian system that I'm writing here because the dependencies with the parents are linear. And we have some noise that could be Gaussian, and that's called the linear Gaussian system. In the context of causality, when we have intervention, so here, for example, I intervene on node B. This is an example of a perfect intervention, meaning that I completely remove dependency from B to its parents. And so B does not depend on A anymore, but B is just some other exogenous noise term. And actually, there are some other noise models that exist. So we've talked about those shift in the soft interventions. Also, imperfect intervention, I guess, is a kind. Soft intervention is a kind of imperfect intervention. And so it's not clear if this is the perfect model for fitting this type of data. But this is what I've worked on for this talk. So I will assume perfect interventions in this context. There are different ways of fitting causal graphs. The first one is constraint-based methods. Those methods will basically search for DAGs by looking at those the conditional independence criterion that we've seen before in the primer. So you need an algorithm for kind of exploring all of the conditional independence tests. You need to condition a specific subset of the graph, and there are ways of doing this kind of searches. But it quickly gets combinatorial with a number of nodes of genes. So this method is not really applicable to a large graph. The other ways are a score-based method that try to turn the inference problem into an optimization problem in the space of DAGs. So then it gets, as we've talked, it gets complicated. How do you parameterize DAGs? You can parameterize them based on permutation, and you can have some nice geometry. So there's really nice work done here about the geometry of permutations and the space of permutation and how you can optimize over that. But it still gets difficult to have really large, to work with large graphs. So there is something that somehow there is another way to think about it, which is parameterizing DAG inference as a continuous optimization problem. So it doesn't work for arbitrary type of like models, but in this context of a linear Gaussian system that I've shown before, it can actually work. And so this method called no tiers provided a way of casting this inference problem over DAGs into a continuous optimization problem. The approach works as follows. So first, you need to define a likelihood model. So here, my likelihood model is just every gene can be a function of all the other genes, and it's a linear function plus some noise with equal variances across all genes. I'm adding some sparsity regularization on the weights of these regressions. And we want to solve a constrained optimization problem, meaning that here W is my causal graph somehow. If there is non-zero value, there is an edge in my graph basically. And I want to make sure that this W encodes an acyclic graph. And I will explain, so there is some kind of function C that takes as input this weighted adjacency matrix W and kind of returns 0 if it's acyclic and non-zero if it's not. I will give you an intuition of how to build those type of penalties. And it's still a topic of active research. But basically with this setting, we can solve this optimization problem with stochastic optimization using an augmented Lagrangian method. And this was what this paper introduced. So I want to quickly talk about yes.
So in blue, can you explain the intuition for trying to recover xi by applying W?
So yeah. When you write the SEM, basically you can see that you do topological sorting, and then you start from having some exogenous noise, and then every node will be, the value of every node will be a linear function of its parents plus some noise again. And so somehow when you fit this matrix, you have a consistency, like you can actually recover if the true model is linear, you can recover the right model.
So would W being the identity, would that solve this optimization?
So W has to be off diagonal. You cannot have self loops. I didn't write that on the slide. Yeah. Actually, the model doesn't learn anything. It comes up in the talk later when I do those factor graphs. I need to remove self loops explicitly, otherwise the method fails for the reason you just mentioned. So I want to give an intuition for the acyclicity characterization. So if A is a binary adjacency matrix, and we don't work with binaries, but somehow I can explain there is just a trick, but this is important to build intuition. So if we take the binary adjacency matrix of a directed graph, we can take power, the matrix power of that adjacency matrix, and at entry ij, we basically are counting the number of paths that are connecting node I to node j in length exactly k, because we're just summing across all the possible neighbors, and we're just calculating those values. So and once you see this, basically you just need to understand that the matrix exponential, you can think about it as a Taylor series expansion. And when you look at the diagonal values of this Taylor series expansion, you have 0 values, or I guess 1, the first term is identity plus the sum of the power. You get either 1 in the diagonal if you have no cycles, or you get something non-zero if there is a cycle. a path from I to I. And for this reason, the first notiers propose that penalty or that characterization. So for a binary adjacency matrix, you can calculate trace exponential of the matrix minus the dimension. That sums all the diagonal values. And this is 0 if and only if the binary matrix A or the graph, I guess, has no cycles. Many other penalties have been proposed. And when you have a weighted adjacency matrix, you would take either the Hadamard product with itself to make those things positive. If it's positive, it still works. It just doesn't work when it's negative. And yeah, there are a lot of discussions about this, but this is basically just giving a snippet of like what this acyclicity characterization is. So it's a numerical characterization for acyclicity. This is differentiable, and you can fit this with first order optimization. So many extensions of those methods were proposed for going beyond the linearity assumption as well as handling interventions. The scalability remains a problem. So the acyclic, this trace of exponential it takes a cubic time to approximate to compute. The gradient as well, it takes a cubic time. So that's what we're interested in, and it's kind of problematic. Doing a matrix inversion with matrices that are bigger than 10,000 by 10,000 at each step of stochastic gradient is a huge problem. And also the existing nonlinear likelihood model can have a lot of parameters. For example, every gene is a neural network of all the other genes. That gets quickly complicated. You run into memory issues, and you run into, you can obviously overfit, etc. So we need to, in this context, to think about what we can do better. And to address those challenges, we came up with this method called DCD-FG. It's a lot of consonants. I don't think it's the best naming, but it looks a bit aggressive, I think. But basically, D is for differentiable, CD causal discovery, and factor graphs. So differentiable because we can solve this problem with first order optimization. Causal discovery, we fit this causal graph. And factor graphs, that's kind of the main innovation here that I haven't talked about yet, is the fact that genes cannot arbitrarily be connected one to another, but they're connected through these things called factors. And I will introduce those things. So we're going to factorize those graphs, and I'll explain what this means. And the point is that why it's interesting is that after we use those factor graphs, the complexity for the acyclicity penalty drops from cubic to linear. And there is a prefactor, which is, of course, the number of modules of factors that you have. And so we're going to be able to scale up this causal discovery method. Any questions on the premises?
Envisioning, well, I guess, are you fitting this on observational data or
on both? On both, yeah, of the rational data, and we put all the data together. So I think in the paper, actually some reviewers asked us, and then they asked us many things. I think they asked us to run on only observational data, and we did that somewhere in the appendix of the manuscript. But when we thought about this, I mainly was excited about using it for perturbative data. So I always had in mind, I want to work only with a lot of interventions and enough to like uncover most of the graph. But I think at some point we have some results in observational data somewhere. And so the reason I've shown this no tiers with a very simple model with no interventions was that because it's easier to understand on the slide, but it's easy to change this objective function to take into account the interventions. All right, so let's get into this. So I'm going to start introducing those factor graphs. So first, I want to build intuition for why we need those objects and what they mean. So I'm going to introduce this object called factor digraphs. The key idea is that this is this is really a central point, which we've been talking about in the primer as well. The space of DAGs is really not nice to work with. And the point is, can we restrict ourselves to a nice subset of those DAGs that can have good properties for inference and good computational efficiency? So here is on the right a gene network from V1 to V7. We have 7 genes. We can see how they're connected. And I'm telling you that somehow this is one of those gene network with nice properties. So this is not clear from this, but the key point is this gene is, so this graph is special because it's part of a special class. I'm going to explain how to get this graph, but basically we can, instead of focusing on how every gene can be arbitrarily connected to any other gene, we want to bias the connection to go through those factors or those modules and how we have those connections. So here, actually, the reason it's a special graph, it's because it is kind of like the collapsed version, and I'll explain exactly what that is. This gene network is a collapsed version of this factor digraph with 3 factors here. And the key idea is that those factors or those modules, I will be using those words interchangeably. They correspond to a unique set of parents node in the gene network. Okay, so F1 has for parents V1, V2, V3. It means that if a gene downstream depends on V1, except if there is another factor, but it has to depend on V1, V2, and V3 at the same time. Okay, so basically when we reconstruct genes downstream, we need to pick those from this unique set of parents, and we can combine them, but we need to pick those modules. And biologically, we hope that those modules can correspond to protein complexes and pathway and gene programs. And I will show the best interpretation I have on the real data later. We'll see that it's not too clear, but even how to, but at least this is kind of like the motivation for having those factors here. So what is a factor graph? A factor graph is just a bipartite directed graph. So a bipartite graph usually means that you have 2 sets of vertices. Here, the genes and the modules, or the features and the factors. I'll be using again those words interchangeably. And what characterizes a factor graph? It's simply that a gene can be only connected to a factor, and a factor can only be connected to a gene, but we can't have gene-to-gene relationships or factor-to-factor relationships. So when you look at the adjacency matrix of this graph that I've shown here on the right, the diagonal of block 0, and we only have off-diagonal values. These are called the partial adjacency matrices. And basically, we can, on different off-diagonals, you have either the ingoing edges from genes to modules, or the outgoing edges from module to genes. Okay, so, and let's try to remember those colors because they kind of matter for the algorithm later. But so orange and green edges. And so then an important question is, again, what does it mean to collapse? So how do we get back to an original causal graph? So we have the big graph that's called the bipartite graph, I guess, and we can collapse it using this notion of half square. So we can calculate any bipartite graph induces 2 half squares, an half square over each of its reduced set of nodes. So for example, an half square over the modules and an half square over the genes. And the half square over the genes look like the graph with the salmon color on the right, the top one. And this is exactly the graph that I was interested in to begin with. And the bottom one is also important. It's a half square over the factors. And it's important for the algorithm part a bit later. But these 2 things are defined. I just want to say that those things are not unique, meaning that if you start from a half square, there are many different graphs that could give you the same half square. Which means that there are different ways of parameterizing some of those causal graphs.
I have a quick question about the way that you view, I guess. I mean, if you interpret these as sort of like latent somehow programs factor, something like that.
Yeah.
But I guess in Jiaqi's framework, everything is downstream of the factors, but here some of the genes are upstream of some of the factors. So can you just maybe?
Yeah, it's interesting. I think those point of view are a little bit different. Like for me, I put the exogenous noise at the level of the genes, while what we've seen in the primer, the exogenous noise is in the latent variables. So to me, I will introduce it will be clear when I introduce the model later. But somehow I'm going to attach to every factor a deterministic variable, which is a function of the genes upstream. But I'm not going to add noise there, but I'm going to add noise downstream. So I think there is definitely some kind of connection. If not now, because now I'm just talking about graphs. When I start talking about the model and I don't really have to, but I can attach deterministic values to those factors. And then it kind of looks like a latent space, but not exactly. And I've never even visualized it because I wasn't thinking about things this way. So yeah, it will be clearer to answer your question when I go back to the algorithm. But I hope that it still already answers it. Yeah. So, all right. So we have those 2 half squares. And so why does it even make any sense? one thing that I thought was quite interesting is that when we look at the adjacency matrix of the half square of a graph, we can calculate it. So it's a binary matrix, right? And we can recover it from calculations from U and Vs. But because those are all binary matrices, if we just do some linear algebra, we get values like 1, 2, 3 that will count the number of paths somehow. But if we look at the Boolean algebra, we exactly get this product decomposition. So this is actually some kind of a linear algebra for the Boolean arithmetic, and which means that the half square of the causal graph that we get, the half square of this big graph, is somehow mean that we're looking at low rank approximations of the gene network. That's why this is kind of an appealing method or nice way to think about it. So we're doing approximate causal inference by like looking for the nearest Boolean low rank graph. Okay. So and then the paper. So the paper that I'm presenting today is mainly a theoretical paper. There is a lot of statements. I mean, and I mean theoretical meaning that doesn't have a lot of application to perturb-seq. Only one I will talk about today. But so the content is emphasized on how does a computational speedup comes along, and I'm going to give you some kind of brief intuition for it. How do we get, what are the exact connections to Boolean low rank decomposition into other low rank models? There were a couple of existing low rank methods for this context, but they don't always mean the same thing. The Boolean rank is not the same, is not at all related to the linear matrix rank. So there is a lot of like different things here to kind of consider. But yeah, I will stop here. We talk about identifiability, and we talk about some kind of resilience to noise. And if you don't observe, if you don't know the skeleton of the graph and you have some errors on it, how stable your kind of class is. But I'm happy to talk about those at the end of the talk. I have slides after this. So instead, for the remainder of the time that I have, I will talk about the model and the inference procedure and the simulations and real data. So, okay, I've introduced this object, f-DAGs, which is actually pretty simple somehow. So what is less simple is how to apply this to causal discovery. So to do this, we need to follow the recipe. First, we need, that I've shown you before, the no-tiers type of framework. So first, we need an acyclicity score, and we need to parameterize the graphs. Then we need to get a likelihood model. And finally, we need a paradigm for optimization. This is something, again, I have another slide, but it's basically derived from another manuscript. So DCD-I haven't introduced it, but they basically took the no-tiers framework, added a bunch of neural nets, and also dealt with interventions. And I'm using exactly this kind of loss functions that I have again in other slides. So how do we parameterize the graphs? So again, we're going back to the person's question. Okay. Yes, if you have self-loops, the model doesn't work. So if you want to parameterize the graph as matrices U and Vs, and those matrices are somehow independent, sampled from some Gumbel sigmoid or some of those continuous relaxation of binary distributions. When you do the product U and V, you get a lot of self-loops, right? Because very often you will have a gene pointing to a factor pointing to the same gene. And with those self-loops, the learning method doesn't work, right? So we need to find a way to parameterize this to prevent self-loops. That's the purpose of this slide. So in this setting, U and V, we design them from a third, they are deterministic function of a third matrix, W. W basically tells you is of size number of factors by number of genes. And it tells you between gene I and factor J, is my edge an orange edge, like outgoing, or is my edge a green edge ingoing, or is it no edges? Okay, no edge at all. So we have three, it's a category, we parameterize it with a categorical variable, like the Gumbel softmax. And this allows for doing stochastic optimization, and it also allows for having no self-loops. And it's been kind of the easiest way for us to get this model to work. Otherwise, the parameterization just collapses. So then the other thing is, how do we characterize acyclicity for those digraphs? So something that's really strong about these factor graphs is that and those, but it's just a property of bipartite graphs, I guess, is that the big graph is acyclic, big meaning the bipartite graph. That's acyclic if and only if the half squares are acyclic. I mean, it's even stronger than that if and only one of the two is acyclic. So this is just an example, just to realize what's happening. But if you have a bipartite graph like this from F1, V1, F2, V2, and you do this half squares, basically you have a self loop between. So here you have a self loop, right? It's not self loop. Sorry, you have a loop. So a cycle. You end up with having a cycle from going through F1 to F2 and to F1 back again, and you have another cycle going from V1, V2, and vice versa. If you have a cycle in one of the small graphs, then there must exist connection in the big graphs, and you get a cycle in the big graph. So that's kind of basically how it works. And for this reason, it's interesting because we have a choice here. So either we calculate acyclicity on the bipartite graph or on the smaller graphs, each one of them we can pick. And when you look at the complexity, so you need to first assemble the matrices, and then you need to calculate acyclicity. So acyclicity is cubic, as I've shown before with the trace of the exponential. So if we take acyclicity on the factor half square version, the complexity becomes m cubed plus m squared d. But if we look at acyclicity on the gene network, we get d cubed plus d square m. And let me tell you that when m is 40 and d is 1,000, you know which one to pick. But then when actually m is much smaller, you get m squared d. You can remove this m cubed part. And there is even another characterization of acyclicity that allows you to get m times d that I will not talk. It's about spectral values and decomposition, but I won't talk about it today. So then now that we know how to calculate acyclicity, we also need a design for the conditional distributions. So we designed a factor likelihood model. So actually, to begin with, I didn't think I needed I didn't really think when I was building this model about reusing the factors. To me, the factors were just an artifact sitting there for the purpose of having a low-rank graph. But of course, you can't have just, if you have 1,000 genes, you can't have 1,000 neural networks. You need to think about a way of sharing those parameters. And it was not super intuitive for me on how to do it. Or you can just have a linear model, and that's fine as well. But I wanted to build models that were not linear. They were already one method actually called no-tiers low rank. I will show a bit later. And so one way of doing this is, so we have those factors anyway, they're sitting there in the graph. So can we use them in the computational graph as well? And that's when it starts to make sense to think about representations. Even though, again, this is not something I've explored in this context, but that's where the connection to me is. So I need to build, again, this likelihood model. So I want to decompose every gene must be a function of the parent's gene in that graph. And again, the parents can be obtained by looking at upstream with a V matrix and then upstream with a U matrix. And then you're free to pick how you build up this function. So the way I did it is I call that a factor likelihood model. So for every factor, you attach a deterministic variable that is basically collecting all of the upstream genes and combining them with a neural network into that single value. And somehow this is, if you think about those Fs as gene modules, it's kind of the activity of the gene module. And then to look at downstream a gene, every gene will collect with a linear combination all of the upstream factors. So when you get into this, it's nice because you don't have 1,000 neural networks anymore. You have 40, 20, 25 neural networks, which is much more reasonable. And you have a model that's nonlinear. So then I did some runtime analysis. So here, in this context, what I did is looking at the time for a backward pass on the full loss function, comparing a couple of different methods. As a number of factors is fixed to 40, and then the number of nodes grows. And so what you can see is I've put a bunch of methods here. Some of them are linear, some of them are nonlinear, and some of them have neural nets. But basically, the remarkable points here are no bears because it's one of the best method. Norbert actually introduced the spectral characterization of acyclicity that is quadratic and not cubic in the number of genes. And you can see that it behaves really well. But by using the module characterization, after 1,000 nodes, we're able to be much faster than them for calculating this acyclicity score. Then you have so you can see that, yeah, basically no tiers low rank or no tiers. No tiers low rank doesn't really exploit the low rank actually assumption for computational reasons, which they could have, but they didn't. So it scales as well, the same thing than no tiers, and it's pretty poor scaling. DCD-FG after is our method with neural networks. And you can see how the only other method with neural nets is DCDI. And DCDI after 500 nodes, I think, basically run into memory issues and stop running. But it's really slow anyway in the first place. And so DCD-FG takeaway is that it's the only nonlinear method that can scale to more than 1,000 nodes on this graph. Okay, so then if we get to how do we evaluate the performance? If you have a fast algorithm, it doesn't work well, it's kind of a problem. So you need to show that the algorithm works decently. So how do you even benchmark those methods? So we took those benchmarking method from the DCDI paper actually, and a lot of other papers before. But specifically, we looked at two things. The first thing is we can do some simulations, and then we know what the ground truth graph is, and we can count the number of edges that are wrong and the inversions and all of this kind of statistics between the ground truth graph and the learned graph. And I will explain how we do this a bit later. And then we can, if we have a lot of interventions, we can start holding out interventions, and we can compare the model goodness of fit on those held out interventions. So, and I will explain exactly what it means later. I just want to say that we do not generate like new interventions. We need the counts for like looking at how good the conditional distributions are. Okay, so we kind of we use the manipulated density that decomposes as every gene depending on its parents, and we look at this sum and we see locally in the graph how the model is while imputing this kind of conditional distribution. Okay, and this is essentially, it makes a lot of sense for comparing, for example, across DAGs, because DAGs are very restricted in how many parents they can take because things are acyclic. And that's what we did in this paper. And we took that from the DCDI manuscript that also looked at this. They wanted a more scientific example of like imputing, and that's what they did. And we did the same. And that we can do on both real data and simulations. So this is kind of the information for the simulation. So we have those data sets with perfect intervention. The targets are known. Each data set has 100 nodes, 5,000 data points. Things are sampled from interventional distribution using a linear mechanism, means a linear structural equation model that I've shown you before, or nonlinear means that you have a neural net with some hidden layers. And the graph is sampled from an Erdos-Irani random directed graph model with factors. We assume that the data is generated from a factor graph already. And we have 100 interventions, and we take 20 of them out for the scoring, the holdout for model evaluation. So the results look like this. We look at two things again. On the left, we have the fitting the held out interventions. On the right, we have a precision recall, like the quality of the graph. And so in this this is a linear benchmark. In the linear benchmark, you can see that two methods work well, DCD-FG and no tiers low rank. Actually, in terms of median, no tiers low rank is better. It looks like DCD-FG is a bit more unstable. So, and the other methods don't work at all. These methods have never been really evaluated on more than like 50 nodes or something like that. So that might explain the discrepancies. And basically, what's kind of so yeah, what is a way to explain those results is that DCD-FG uses neural nets, and no tiers low rank is actually a low rank formulation of no tiers, which is exactly how the data was generated. So it kind of makes sense that if you use a linear model, you get slightly better results. Then the things start to deteriorate when you look at nonlinear. So when you have a tiny neural network for each conditional distribution, you start getting into trouble, and DCD-FG works much better, even though the performance gets down for a lot of different, across all the models. And this is with Gaussian noise, but the reviewer asked us to do with less interventions and with uniform noise, and I forgot everything, but yeah, it's in the manuscript. One thing that's important is how do you pick the number of factors? I don't have a slide for that, but basically, and it's an interesting question in general when we do causal representation learning, like can you show that your method is robust to the number of factors, or how can you have an algorithmic way or greedy way of picking them, etc. So here what I did is just, I used 10, 15, 20, 25, and I did some kind of grid over 40, and I look at, I have a validation set of interventions, and I use this interventional NLL. So it's not the same interventions, but it's just some I can hold out and use that for scoring my models. And you can see that the model with the worst likelihood really just do not perform well in terms of graph metrics as well. So those things seems to be correlated, even though it's probably not an optimal way of picking those numbers. Okay, and then we applied this to Perturb-seq data. So we applied it to this data set. So we have melanoma cells with expressing Cas9. Cas9 is a CRISPR-Cas enzyme that can cut the DNA, introducing double-strand break. So we have also a library containing 248 or 249, I forgot actually, number genes. These genes are immunocancer resistant genes, and we have different environments. So either the cells have been, or control cells are just resting, or the cells are treated with interferon gamma, or the cells are co-cultured with leukocytes and for 48 hours. And then we perform this assay I've showed you, Perturb-seq. The reason I picked this data set is because there are 3 environments. And so I thought, oh, it's for one data set, I get 3 set of results. And also it was one of the most recent data sets when I started working on it. It was pretty exciting. Again, there are around 200,000 cells. So you see, even if 200 genes is not that much, there are already 200,000 cells with the 3 environments. So like the scalability quickly comes around. The goal of this experiment was to identify gene networks that confer resistance or sensitivity to T-cell mediated killing. Then you need to construct a set of genes. So what I did is picking highly expressed genes and adding all the genes that we had perturbed, because I need to have them in the network. Otherwise, I just don't have perturbations. And in every data set, I basically take 20% of the interventions. I take those cells out and I use that as a test set to simulate what I had from the previous data set. And so the result looked like this. So we have the interventional negative likelihood, which is exactly what I've shown you before. And because here it's a Gaussian model with unequal variances, I learned the variances. So basically it's kind of like, it's a square, the mean square, it's a weighted mean square error by the inverse variances. And so for having another metric, I calculate the MAE as well. So if it was MSE, it would be just the unweighted MSE, but here it's MAE. MSE looks the same, honestly. This is on one data set, and you can see that the method has much better fit. And it's the same thing for all the 3 data sets. Any methods or any questions? So then I want to show you how I was kind of interested in, okay, where can we push this, and what is this graph? What does it look like, and is it interesting? And so in this context, what I've done is, the graph is basically 1,000 by 1,000. It's like huge. So you look at the adjacency matrix, and quickly you figure out it's going to be rough to just make sense of this. So the second approach was, I have been working on all these half squares. So how about I just plot the half square over my modules? So here the best number of module was 20. So I have, you see, those numbers are basically designating the labels for each of the factors. 20 factors. Okay, let's plot. So the gray things are somehow when it's like the skeleton of the half square. Okay, so I can do some topological ordering because I know it's a DAG and it looks like a tower. So first, it's interesting because it looks better than just a bipartite graph that we had from previous Perturb-seq type of experiments. So it looks like it's learning something. Then we want to figure out what. So one way to do it is I look for, in every module, we have genes coming in and genes coming out. I can look at what are those genes, run a gene set enrichment analysis, and just color the nodes. So this is how I get those colors. Okay, and things look relevant to cell cycle, antigen processing, and yeah, like so some TGF beta regulation. So all of these things are kind of relevant to the genes that were the gene modules that were picked up in the original publication. Still not that great. Like you can't really make sense of a lot of things if you just had those colors. So what I tried to do was point out a few genes that I know are supposedly important in the context of this paper. So I picked some genes that are supposed to be the most effective perturbation, and I wanted to place them on the graph. Those are the red genes. And same thing, I looked at the most important genes that were actually written down in the manuscript, and I picked them, I put them in the graph. So how do I put those genes? It's not clear, right? Because it's a half square over the modules. And there can be many edges. So the same gene can be upstream and downstream of several factors. So I need to pick the most important one. The way I did this was just looking at the strength in the network of like, so the very simple thing. And I obtained that graph. So that's how I get the graph I've shown you. And then you can start interpreting. So, I mean, in a limited fashion, honestly, but so we can see that Jiaqi points to interference signaling and to STAT and a lot of HLA genes. On the right part, we can see CDK6 points to HLA genes as well. And in the middle, interferon gamma receptor 1 points to other interferon signaling genes. And there are some other things that make less sense. For example, there is a lot of TGF beta that wasn't really mentioned in the original paper. And you have JAK1 and JAK2 that are supposedly working together from as far as we know, but here they look in completely different part of the causal graph. So I don't know if it's just my method of like attributing things in the graph that is not great. Maybe it appears over there as well. I just haven't looked really into it. Or maybe it's just the graph is not that interpretable. And I want to give some closing remarks. So in this talk, I've shown you the usage of half square of factor graphs in the context of causal inference and some of their theoretical properties. We've developed DCD-FG, which is a tractable model and inference procedure for scaling up causal structure learning on high-dimensional data sets. We've applied it on simulations as well as perturb-seq data. And the direction, so we were really interested in making this work on cyclic graphs. And we have some work basically on, it was so much work to have any model that is just like kind of half scalable on cyclic graphs that we didn't get to using it with the factor graphs, but this is a manuscript that I've put there. The key insight about when you think about cyclic graphs, we were talking about times, etc. So I'm just going to take a minute to talk about this, and we should delay questions to discussion offline. But basically, when you look over the cyclic graphs, you want to think about it as a fixed point. So one semantic is you have some exogenous noise vector that you sample, u or epsilon, I guess epsilon, and then you want to find a fixed point x equal, I guess if it's a linear model, ax plus epsilon, and then a can have cycles, but it needs to have some property, like for example being contracted. I could do this on the whiteboard later, but and under some assumption you can relax this to be not linear but to be a contractive function, like a function with Lipschitz constant strictly less than 1. And then you can parameterize this with flow and you can start making inference, and that's what this paper is about. But I don't think we can use interventions as well. I just don't know the theory then of like what this identifies or what this means is pretty finicky. And that's everything I'll say. But besides doing that, there is also a lot of work that needs to be done on like what are biologically informed evaluation metrics for improving the model. Like once you have those graphs of gene by gene, how do you score them? So there are some recent papers. I'm not too satisfied. I mean, it's good. We need to have starting points, but I don't quite know what is the right way of doing that yet. And we need to include, it's important to think about as well prior knowledge. Maybe the problem that we're tackling here is already indetermined. And so if we had ways of like constraining the model from some prior knowledge, we would be better off using that for the purpose of having even more interpretable inferences, etc. And then finally, I've shown you this first bulk at analyzing one of those graphs. And it looks like even showing this to a biologist would be pretty complicated to prioritize what to test, right? There are so many hypotheses in there. And so somehow, yeah, finding a streamlined way, or like a pipeline way of doing that would be pretty important for this kind of work to shine. Anybody who will come up with the causal graph will need to spend time thinking about what is actually worth testing, what is worth investigating, what kind of assay do we have to check those hypotheses, etc.